Okay, hi, good afternoon. Let's get started. We will start by doing some questions from the study guide, just as a review, right, of some of the key things that we've learned from last class. Okay, specifically, we're gonna review the carbohydrates portion. So if you have the study guide, please take it out. If not, then that's okay as well. Just write this down somewhere in your notebook, and you can fill this in at a later time. So we were talking about carbohydrates last time, which is one of the 4 types of macromolecules that is important in a living organism. And when we talk about these things, we want to pay attention to the elements that it is made up of, as well as the monomers and so on and so forth. So let's fill this in together. Okay. For carbohydrates, the elements that it contains include carbon, hydrogen, and oxygen. The building blocks or the monomers of carbohydrates are collectively known as monosaccharide. And there are 3 monosaccharides that we discussed last time. Does anybody remember what they are?
Glucose, fructose, and galactose.
That is very good. Okay, thank you. Glucose, fructose, and galactose. Okay, so these are monomers. You can join them together and build a bigger molecule. In terms of biological function, we use carbohydrates for fuel, for energy storage, as well as for structural support. Right? So those are the key things that we should know about carbohydrates. Now in this diagram, we see 2 glucose molecules. And if we want to join them together, we will be removing an H from one of them, and then we'll be removing OH from the other. And then we're going to be able to form a new bond that connects them together, right? So I'm not going to ask you to draw things on the test, but it's a good idea to, be comfortable with how these things are formed. And you don't have to draw the whole thing, but once we connect them together, and we get something like this, right? So this O right here, that's like this O in the middle. So here we have a covalent bond, which, as you know, involves a sharing of electron, right? Sharing of electron. And over here, if you take a glucose plus a glucose, you're gonna get a maltose. Okay. Of course, because this is a dehydration synthesis, we are also going to be producing H2O. Okay. That's where the H and the OH has gone to, right? They come out and then they connect, and you get your H2O. So let's try to fill this out together as well. To get maltose, we need to have a glucose plus a glucose. How do we get sucrose? Does anybody remember? What are the 2 things?
Glucose and galactose.
That's right. Thank you. Glucose and actually, galactose is for the next one. Yeah. Maybe
Fructose.
fructose. That's right.
Sorry,
Very good.
fructose, yes.
Yeah. And then you have glucose and galactose, right? Okay, so sucrose is like kitchen sugar, right? Lactose is found in dairy products. And I just want to remind you, all these things over here, all these things are monosaccharide, single sugar, right? And all these stuff over here, they would be disaccharide, double sugar. Okay, this is just a review. Okay, we're not learning anything new. Now we can connect multiple sugar together, and we're gonna end up having a polysaccharide. So an example of polysaccharide would be starch, and we will find it in the stem or root of plants, right? And the plants use it for storage, right? Energy storage. For plants. Okay. Glycogen is another example of polysaccharide. We would find it in animals, including humans. And does anyone remember where in the body will we have glycogen?
Muscle?
That's very good. And another place? What's the organ that stores?
Liver.
That's right. Muscle and liver of animals, right? We'll just say humans, okay? Humans. But other animals too. And the function is energy storage. For the body. Next, we have cellulose, which is the fibers, right? Fibers in plants. You also find it in cell wall of plants. And the main biological function is to provide structural support. Last but not least, we have the chitin, and you will find it in the exoskeleton of things like crabs, right? Anything that has a shell, lobster, insect in some cases, if they have a hard exterior. And again, it's to provide protection. That's all. That's everything we learned about carbohydrates. Does anybody have any question before we move on to lipids today?
So we said polysaccharide is the same of polymer or
Okay, so
I'm
polymer
confused.
is just a general term. Polymer means very long molecules that have repeating units. So when we talk about carbohydrates, the type of polymer that it has is called polysaccharide. Right? When we talk about other macromolecules, like when we talk about protein later on, the polymer for protein would be called polypeptide. Right? We talk about polyester last time, right? That's an example of a polymer. So polymer just means it's just like a broad term for very long things. And there are different examples of it. Is that okay?
Yeah, I got it.
Very good. Okay. That's it for now. Let's move on to lipids, all right, in the PowerPoints. So lipids, lipid contains carbon, hydrogen, oxygen, just like the carbohydrates, right? But it also have trace of phosphorus for some type. Right? A little trace means a little bit, right? A little bit of phosphorus for some of the lipids that we will see. Now, lipids is a little bit different compared to other macromolecules. We already learned last time that the macromolecules primarily are going to be like this. Right? It's going to be like a bunch of subunits that are connected together, right? But for lipid, it's a little bit different. Okay? For lipids, rather than having the same unit over and over again, lipid is more like they have different components. So like there is component A, maybe there is component B, and then maybe there is like some component C. And when I want to build a lipid, rather than having, a bunch of A's connected, like A, like that, we pick and choose the component. Maybe we take one of component A and then we connect it to 3 of the B's, okay? And that's going to be a lipid. In another situation, maybe you take an A and then you will connect to 2 of the B's, and then you will connect it to a C, and then you would have another kind of lipids. So it's not really a polymer in the sense that it is multiple units connected together. Right? Rather, we have different parts, different components that are available, and then we just kind of assemble it together to make a lipid. Okay, so this is class notes. I don't think we're going to be writing too much to it. We'll see. But that's the main difference. Okay? And all lipids are going to be hydrophobic. We learned about this term last time, right? Hydro means water, phobic is to be afraid of. So they are afraid of water. They do not dissolve in water. What's the opposite of hydrophobic? Does anybody remember from last class?
Hydrophilic.
That's right. Hydrophilic would be water loving, right? That they are able to dissolve in water. So let's take a look at different types of lipids, starting with fat. Fat is also known as triglyceride. Triglyceride. If you do your body check, your doctor is going to look at your blood and check for the triglyceride content, right? And tells you if it's in an acceptable range. And the fat is made up of 1 glycerol and 3 fatty acids. So that's a component, right? The glycerol, it's like component A, and then you have like B. That's what I was trying to draw in the picture, right? Now, to connect the glycerol to the fatty acid, we will be doing the same kind of process as we did in the carbohydrate. We are trying to remove a water molecule. So you take away the H here, you take away the OH, and you form a bond. Right, so that's the bond, that's the bond, that's the bond. And because we are trying to connect 3 fatty acids onto the glycerol, this process is going to generate 3 water molecules. Okay, so glycerol plus fatty acids, 3 fatty acids. All right, we'll take a closer look at these fatty acids later, but they are primarily just a bunch of C's connected to H. You can see some of them are longer, some of them are shorter. Some of them have what we call a double bond, and that is going to change the property of the fat. And we will talk more about it in the upcoming slides. Now, there is no shortage of lipids, of fats in our food. Most of the food we eat is going to be gonna have some fat. Fat is not necessarily a bad thing. Okay, we need fat in our diet. There are good fats, and then there are the bad fat. Right? So the good ones are usually. Anybody know the names of any good fat? Usually they.
Avocado.
Yes, yeah, very good. Avocado is usually considered as good fat, but like I'm looking for like a specific name. Usually they would write it on your eggs, huh? Like these kind of good fats.
Omega.
That's right.
Omega
Oh,
3.
Omega 369, right? Those are the good fats, right? So things like salmon, seafood, and even nuts. Some of the nuts they have very good fat content, the good kind. So you know, those you can have a little bit more of, and it's going to be okay. And then, of course, there are those bad fats, right? Fats like butter, those from bacon grease, all those things from chips, right? Those are not necessarily the good kind of fat. So the fat is made up of, once again, 1 glycerol and 3 fatty acids. The fatty acids can differ in lengths, as you saw, but it can also be different depending on how much hydrogen it contains. If it has the maximum number of hydrogen atoms already, if it is saturated with hydrogen, then we call it a saturated fat. Okay, so let me go back to the other picture here. You can see, and you will learn this in chemistry later on, okay? But when carbon connects to other atoms, it can only make 4 connections at most. Right? So you can see each of the carbon in the chain is already making 4 connections. 1, 2, 3, 4. It has no more room for any more hydrogens. Okay. In other words, it is saturated with hydrogen. So if all 3 of the fatty acids are saturated fatty acids, then the whole fat is going to be called saturated. Okay. So here is the glycerol. And then here are the saturated fatty acids, okay? Again, saturated just means they have no more room for additional hydrogen. Now, if I put some saturated fatty acid next to each other, they have no problem getting real close with one another, okay? Because they don't take up a lot of space, right? So they clump together easily, and that is why saturated fats are usually solid at room temperature. Most animal-derived fats are going to be saturated. Okay, so butter, lard, right? Lard, bacon grease, okay, all these things are saturated fatty acids. The problem with saturated fats is that they clump together easily, and so they might clump together in your arteries, and that could cause health problem, right? Cardiovascular diseases. So you would want to avoid saturated fat in your diet. But sometimes if you want to put butter on your toast, then you got to put butter on your toast, right? Like you don't necessarily think about these things. The opposite of saturated fat is unsaturated fat. Right? Unsaturated fat, they also have a glycerol. They have at least 1 unsaturated fatty acid. Let me go back here to show you. Okay. So this fatty acid is saturated because all the carbons are bound to as many hydrogen as possible. This is also saturated, but this one is unsaturated. Okay, because look at this carbon. It only has 3 things, 3 connections. 1, 2, 3. So that means it has room for more hydrogen. Okay, when this happens, we have what's called a double bond. Double bond. If you are not sure what a double bond is, don't overthink it for now. Okay, you will learn in chemistry later. But all that means is the carbons have more room for hydrogen. Okay, So whether or not the fat is the whole thing, right? Whether or not the fat is saturated or unsaturated depends on the kind of fatty acids it has. If all of the fatty acids are the pink part here, right? If all the fatty acids are saturated, then the fat is also saturated. But if one of the three fatty acids are unsaturated, then the fat is going to be unsaturated. It's possible to have two unsaturated fatty acids. We still call it unsaturated. And you can also have all three of them being unsaturated fatty acids. And in the case, it will also be unsaturated fat. So because of the unsaturated fatty acid, there is going to be a bend in the structure, a kink in the tail. If there's a kink in the tail, then the molecules cannot come together too closely. The kink is going to be sticking out. It's going to be in each other's way, right? Imagine lining up next to people at the grocery or something, okay? But then like you stick your hand out, right, to keep people away. Right? That's the kink in the structure. There is this thing that is sticking out, right? And it prevents another molecule from getting too close. And so they don't pack together easily. That's why they're going to be liquid at room temperature. Most plant-derived fats are unsaturated and they are relatively healthier. So do you guys have any example of oils at home that would be unsaturated?
Olive oil?
That's right. Olive oil is unsaturated. Anyone else? Yeah, go ahead.
Avocado oil.
Yep, avocado oil, exactly, right? So
Nuts. What about nuts oil? Like
what would be
walnut.
like a not unsaturated? Like coconut oil. Okay, coconut oil is more like a white paste, right? So that would be saturated instead. Okay, so most plant-derived fats are going to be unsaturated. Are we together on this?
Yes.
All right. So there is a table. There is a table in the study guide. Where is it? Right here. Oops. Yeah, there we go. Number 13, right? That you should fill in, okay, to compare the saturated fat and the unsaturated fat. Right? We'll do it later on if we have time today. But if not, you should do it. I'll post the answer, of course, and you can check it as well, right? But we should move on. We should move on for now. Now, some fatty acids are essential. Essential means you cannot make it in the body, and the only way for you to obtain it is through your diet, right? So example would be omega-3, right? And there are different types of omega-3. Examples would be like EPA and DHA. You don't have to memorize those, but if you are if a person is pregnant, usually the doctor would suggest to take these as supplements because EPA and DHA is important for brain development in the baby. For those of you who are interested, why is it called omega-3 and how is it different from like 6 and 9? It has to do with where the double bond happens. Okay, so for omega-3, the double bond happens in carbon number 3. Right? So we count the carbon. Each of these dot is a carbon, right? So carbon 1, carbon 2, carbon 3. If the double bond occurs at carbon 3 for the first time, then it's called omega-3. Okay? Again, you don't have to memorize this. It's just like extra information. So finally, one more time to summarize the difference between saturated and unsaturated. Right? They both have this molecule, which is the glycerol. And then, so that's the glycerol. And then the other stuff, the rest of it, right? Like these here, these are all fatty acid. You can see there are no double bond in the fatty acid. That's what makes it saturated. Over here, we have double bond for all of the tails. So sometimes they are called unsaturated. Okay, as opposed to just one tail is unsaturated or two tails, right? So you can see there is a kink here, a kink, and a kink. Every time you have a kink, a bend, it's because of the double bond. Okay, so saturated on the left, unsaturated on the right, and the unsaturated one is the relatively healthier choice. Moving on. So, even though unsaturated fatty acids are supposed to be healthier, right? But it's not really practical, right? Back to the toast. In the morning, you have a slice of toast. It doesn't really make sense to like drizzle olive oil on it, right? Like it's gonna make it soggy and maybe not taste good. So people came up with a way to convert unsaturated fat into saturated fat. Okay, by adding hydrogen. Right? Remember, it's unsaturated because it still has room for hydrogen, right? So by adding hydrogen to them in a process called hydrogenation, right, you create what's called hydrogenated vegetable oil. So they were liquid to begin with, they were derived from plants, but then you add some hydrogen to it, and now they are spreadable. Okay, so these are things like margarine shortening. But a byproduct of this process is trans fat. Trans fat. Okay, and trans fat is like the worst fat of all time. Our body cannot break down trans fat. If you consume trans fat, it will stay in your body for a very, very long time. And if they build up and there's too much of it, then it increases the risk of getting heart diseases and things like diabetes. Okay.
Vincent,
Yes.
so when we butter our bread, does that mean we have some kind of trans fats in our system?
Okay, so like if you use butter, right? Butter, butter is purely saturated.
Oh,
So
okay.
that's like natural state, if you would.
Oh, okay.
Yeah,
Okay.
it's like the margarine, right? You know, they say, oh, I can't believe it's not butter, right? That's like the slogan, right? So those
Oh,
are
okay.
the margarine, those are the hydrogenated one. Those are the ones that are
Oh,
artificially being converted. Okay.
okay.
However, because trans fat is so bad, um, and you know, it's linked to all these cardiovascular conditions, obesity, and so on and so forth. Canada actually banned trans fat in 2018. Okay, if you want to read more about it, you can click on this link. Okay, so manufacturer can no longer add trans fat to the food. People add trans fat to the food because number one, it makes them taste better, especially when it's fast food. If you fry the chicken or whatever in trans fat, it turns out it tastes better. Number two, trans fat can also extend shelf life. If you're buying something from the supermarket or something and it contains trans fat, it's there to make it last longer. Okay, but you know the government say no more. Okay, you are not allowed to do that. So they banned trans fat completely in 2018. So the food that you eat should not contain any additional trans fat. If it's naturally occurring, then it's okay. You know, they don't make you take those away. But if it's introduced purposely, then you know, it's a banned product. All right. So that's it. I just want to make sure that you are with me in terms of what we are talking about. So I will quickly summarize it for you. Right? We have the lipids, right? The lipids. Lipids. And we only talk about one kind right now, which is the triglyceride, which is the fancy word for fat. Okay, how do we make fat? We have the glycerol plus the tails, right? Fatty acids. They could be saturated, unsaturated. We already talked about it. Now we're gonna look at the second one. Right? Second one. Second one is called a phospholipid. Okay. Phospholipid. How do we make a phospholipid? We need the glycerol again. We need to have fatty acids, but only 2 of them. This one need to have 3, right? And we need a head. Okay. This is called a polar head. Right. Let's go back to the PowerPoint slides. So the second type of lipid, right? Phospholipid. You can see the 2 tails are here. You might be able, it's harder to see, okay? But like that's where the glycerol is. Okay. And then you got the head here, the polar head, right? So the phospholipid has CH and O, but it also contains a little bit of phosphate. Okay, that's the one that's going to show up in the head. Okay, trace of phosphate. Now that makes the molecule a little bit interesting. Okay, so usually we'll draw it like this, and we'll draw a lot of these later on. Okay, it's going to have a polar head, and then they are going to have non-polar tails. Okay? So the head is water-loving, but the tails, they don't like water. This actually is what detergents look like. If you try to wash the plates, right, and it's greasy, if you only use water, you can't really wash off the grease, right? And that makes sense because, you know, water and grease, they don't mix together. We know that already. But when you add detergent looks a lot like this phospholipid. It has a hydrophilic head, right, or a polar head, and then they have the hydrophobic tails or the non-polar tails. So when you add the detergent, the tails will bind to the grease on the dish, and the head will bind to the water. And it's kind of like this intermediary that connects the 2 things together so that you can wash the grease away. Okay? So let me put an extra description here. The head is hydrophilic, and the tails are hydrophobic. All right? So it turns out the phospholipids are excellent material to make up your cell membrane. So let me show you a picture of the cell membrane. We will learn a lot more about the cell membrane in lecture 3 and 4, but basically it's a barrier that surrounds the cell, right? This is a cell, and the cell membrane separates the living and the non-living, right? So all the hydrophilic heads are going to be facing outside or the inside, and then there's a thick barrier in the middle where it's hydrophobic. Okay, the best way to visualize this is to think about a sandwich with with a lot of peanut butter in the middle, okay? So the bread would be like the circle, like the head. Okay, so you have the bread on the outside and the inside, and sandwich between would be the hydrophobic tails. Okay, hydrophilic head. We'll learn more about this in future lectures. But for now, I just want you to understand, to know that the phospholipids are made up, they are the key components that makes up the cell membrane. Next, the third type, we have wax. So wax is basically a water repelling surface. It covers the surface of plants. That's why the water form beads when they are, collected on leaves. The apples, right? The waxy coating of the apple that has some wax on it. We also have wax in our hair and skin that helps us protect it from drying, and it also lubricates them. And wax is also found in like honeycomb, right? People use bee wax for cosmetics, for lip balm, for example, right? So to make wax is basically long fatty acids connected to some alcohol. Alcohol is a broad term, okay? It's not necessarily drinking alcohol. In chemistry or biology, alcohol is anything, any organic molecule that has OH on it, okay? That's what makes it an alcohol. So a wax is basically fatty acids with some alcohol connected to it. Okay, so if we go back to our summary here, number 3 is wax. It's basically fatty acids, okay, with some alcohol, right? Alcohol just means OH. Okay, like some OH here and there. Of course, this is an oversimplification, but that's basically what it is. Okay, let's do the last one here as well before we switch back. Number 4 is steroid. Okay, so steroid is easy to recognize because they always have like 4 rings in their structure. Okay, 4 carbon rings with other stuff that are attached to it. Okay, never mind the other stuff. The core structure is that it has 4 rings to it. Okay, so these are the 4 types of lipids. So maybe just very quickly, the fat is for energy storage, long-term energy storage. Okay, if you skip lunch today, you are using your glycogen reserve, right? From the liver, from the muscle. But if you are fasting for like 2-3 days, then you gotta start burning, your body will start to burn the fat. Fat is a long-term storage, whereas glycogen is more like immediate short-term storage, right? Fat is also good for insulation. Insulation means it keeps the organ warm, right? Around the kidney, around the heart, and so on and so forth. There's going to be some fat there to keep them warm, and it also, cushion the organ a little bit, right? Okay. Cushioning, insulate and cushion organs. That's what it's for. Phospholipid. Do you remember what it was for? Major component of what?
Cell membrane.
Cell membranes.
That's it. Yep. Everybody gets it. Good. Major component of cell membrane, right? Wax is to form water-resistant barriers. Okay. That's why when we wash our hands don't swell up because the water actually doesn't go into the hand, right? I mean, if you stay in the bathtub long enough, then the water will eventually start going in. That's why you have those like wrinkles, right, in the fingertips. Okay, steroid. Steroid has several functions. Okay, we'll come back to it. I want to show you the PowerPoint slides first. Okay, so there you have steroids, the 4 rings, 1, 2, 3, 4, and a bunch of like random stuff attached to it. Never mind those things, okay? Those things are just called functional groups. Functional groups are just group of atoms that gives certain properties. Okay. For example, if it's an alcohol, then it makes it polar. You will learn a lot. You will learn all the functional groups in your second semester chemistry class. Okay. But the key thing, like if I show you a picture and you see 4 rings, then you have to know it is steroid, right? None of the other lipids has rings in it. Right? So the most common steroid that we need is cholesterol. Okay, you might be a little bit confused, right? Because you might have heard that cholesterol is bad, right? Well, it turns out there are good cholesterol and then there are bad cholesterol. And if it's the good kind, then we need them, right? Cholesterol is the raw material to make steroid hormones, okay? And we also need cholesterol in the cell membrane. Most of the cell membrane is made up of phospholipids, but it turns out we need a little bit of cholesterol as well. We will learn why in lecture 4. Okay, but trust me, it's there. And we also need cholesterol to make steroid hormones. Okay, not all hormones are lipids. A lot of them are actually proteins. Okay, but for the steroid ones, many of them use cholesterol as a starting material. So example of steroid hormones are the sex hormone. In male, the sex hormone is testosterone. That's what it looks like. Again, you see the ring structure in the middle, the 4 rings, and then a bunch of functional groups. In female, the sex hormone is estrogen. There is another one. Does anybody know what the other one is called? Starts with a P, huh?
Progesterone.
Very good. Okay, so progesterone, testosterone, estrogen, all these are steroid hormones. And to make them, you need cholesterol. Okay. Again, not all hormones are going to be steroid. Some, as you will see, are proteins. So,
Okay,
yeah.
so Mr. Wang, what are the functions for steroid?
Yeah, so I should just gonna write it here. Okay, so for steroid, it is gonna be part of the cell membrane, and then they also form hormones. Okay, hormones are basically messengers in the body. Okay, like if your brain wants to communicate with the ovaries, for example, they have to send these hormones there. Okay, if your thyroid wants to communicate with the brain, it will also send hormones. Right? You'll learn about all the hormones in the body next semester. Okay, something to look forward to. That's it for lipids. Anybody have any questions? No, we all good? All right, fantastic. Let's take a look at this whole question here, huh? Okay, I try to keep these under a minute. If you could please lock in your choice, that would be great.
Excuse me,
Yes.
teacher. I'm a
Yeah.
newcomer.
Oh.
I'm a new student. I registered on January 15th, so I missed some classes.
That's okay.
And so,
I will show you during the break. I'll show you where to find the missed lecture. Okay.
okay.
Yeah.
And I can't understand and answer this question, and I don't know how to see the record of the class
Yeah.
and how to submit my assessment.
Yeah. That's okay. I will show you during the break or at the end of class. Okay. Just stick around. Right.
Okay, thank you.
Okay. 11 out of 18 of you answered, and all of you got it right. It's the triglyceride. I mean, I've been repeating this for the whole class, right? Glycerol plus 3 fatty acid, you get triglyceride. Triglyceride. Okay, next question. All right. I didn't get as many respond this time, but that's okay. Which of the following is a false about lipids? Number 1, steroid has at least one fatty acid tail. Well, let's take a look at the notes we wrote, right? Okay, steroid is right here. Do we see any fatty acids? Nope. All the other ones have fatty acids, but not steroid. Okay, so the only thing that makes steroid is that it has 4 rings. Okay, there could be other things attached to it, but it doesn't necessarily have to have a fatty acid. So that's why this is incorrect. Okay, everything else here is correct. Okay, everything else here is correct. Okay. And for those of you who don't know, I put the link in the chat box. That's where you can find the lecture recordings, right? If you miss previous classes or if you just want to hear it again for certain stuff that are particularly difficult, I put the link in the chat box. Okay.
Vincent, is canola?
It's canola oil. What? Sorry.
Is canola oil a type of plant? Canola, is it a type of
Yeah,
plant?
canola is the flour. Yeah, they got
Okay.
yeah. So
Oh.
it's unsaturated fat because canola oil is liquid at room temperature, right, for the most part. All right, that's it for lipids. That's it for lipids. We are now going to talk about proteins, and protein is the hardest one of the 4. Okay, there's a lot of things going on in the protein. I will try to Keep it as simple as possible, but please stop me if things are not making sense at all. So proteins, they contain CHNO. And by now you should kind of see the trend, right? All the macromolecules have these triplets, right? They always have CH and O. But for protein, there is also nitrogen, okay? And trace of sulfur, okay? So carbohydrate is just CH and O. And when we talk about chitin, it also has nitrogen, right? For lipids is CH and O, but when we talk about phospholipid, it also has the P, the phosphate, right? Phosphorus. So when it comes to protein, we have the CHON and trace of sulfur. Again, trace just means a little bit of it. So just like carbohydrates, where you connect multiple monomers together, protein is exactly like that. But the kind of monomers are going to be different, right? We call them amino acids. Amino acids. They are the building blocks. They are the monomers of the protein. And there are 20 amino acids that exist in the body. 9 of them are essential. Does anyone remember what essential means? I told you a few slides ago.
The body cannot produce it.
Very good, exactly, Fatima. You cannot produce it. So the only way to get these 9 essential amino acids is to eat them. If you are not a picky eater, then the regular kind of food that we eat already cover all of these amino acids, okay? But they are all listed here. These amino acids listed on the slide are the essential ones. So some food are particularly high in them, okay? So let's say for whatever reason you need to have more tryptophan, then you can eat some oats, some spinach, right? Soybean, sweet potato. Okay, but like I said, if you're not a picky eater, general diet would have a pretty good coverage of these things. All right, so each of these is an amino acid, right? Now, one of the amino acid is called phenylalanine, which is right here, okay? Phenylalanine. Okay, you can find them in avocado, beans, rice, almond, seaweed, pumpkin. I'm not sure why they put the broccoli here because I don't see the broccoli in the list. But anyways, phenylalanine is one of the oh, sorry, the broccoli is for the thing above. Yeah, okay. So phenylalanine is one of the amino acids, okay? And one of the things that they do when the baby is born is they will do what's called a heel prick. Okay, heel prick. So they make a little cut here at the baby's heel and they collect the blood and then they go test the blood. And of the many things that they test for, one of the thing is they test for this condition called PKU, okay, which stands for phenylketonuria, okay, or PKU is fine. So this is a rare genetic condition where the person, the baby, is not able to metabolize, is not able to break down phenylalanine, which is one of the amino acids, right? And these phenylalanine start to build up in the body and it interferes with brain development. So it's dangerous for these babies to consume even the breast milk, okay, because it might contain the phenylalanine. So they have to go on a special diet, right? Special formula, baby formula that doesn't have the phenylalanine. And once they grow up, they have to limit the amount of phenylalanine that is in their diet. Okay, so that's PKU. And if you see they cut your baby's heel, that's what they're testing for, amongst other things. All right, next we're gonna look at the general structure of an amino acid. So let's go back to our class notes here. Let's draw an amino acid. I am not gonna ask you to draw anything. I keep on telling you that, okay? I don't want you to freak out when you see these drawings, but it's a good idea to draw with me so you understand the structure and how things are connected, okay? Even though there are 20 amino acids, they all share a general structure. Right? So there is a carbon in the middle, and it's connected to an H. And then on one end is connected to an NHH. And this part here, that is called an amino group. Amino group. And again, like you will learn all these in greater detail next semester in chemistry. On the other side, it will be connected to a COOH. Okay, we say COOH. This here is called our carboxyl group. Okay. So carboxyl is an acid, amino group is a base. That's why it's called an amino acid. So all the amino acids are gonna have these things, all 20 of them. What makes them different from one another is this, what we call like a side group, side chain. Right? It is different for each amino acid. Okay. Here, we're just going to put in bracket here in case you forgot. These are the monomer of proteins, the building block of proteins. Any questions so far? Okay.
Professor, excuse me.
Yeah.
You have said around 20 amino acid. It means protein just has 20 amino acid.
That's right. And they don't have to be all the same thing. Okay. I'll show you here. Look. These are the 20 amino acids, right? So we'll discuss this in more detail coming up. But basically, think of this as a tray of 20 different beads. You know, the beads to make like necklace, okay? Like arts and crafts stuff. So there are 20 different colors. So you can make a necklace using only 4 colors, right? You can make a necklace using 6 colors, 10 colors, right? It's up to you. You don't have to use all 20, right? And so in the same way, when we build protein, you don't necessarily have to use all of the amino acids. You can just use maybe like the yellow ones here. Maybe you can just use a little bit of here and here and here. You understand what I mean? Does that answer your question?
Yes, Professor. Thank you.
Okay, yeah. So this is a picture that shows you all the amino acids, right? Like, so you see the C here, the H, the NH, and the COO. Okay, but what makes them different is the R group here. Okay, some R groups are non-polar, and it makes the amino acid a non-polar. Some of them are charged. You see, there's a positive charge here, right? Some of them are polar. Some of them are negatively charged. Right? The point is that every amino acid is different in the R group, but then their properties might be similar in that they might both be nonpolar or they might both be polar, so on and so forth. Okay. Now I just want to do one more thing before I go on the break. Here it is. So let's just quickly do 2 amino acid here. So CH and then NHH CO. Okay, there we go. That's one of them, and this is R1, as in like side chain number 1. And I'm just gonna draw another one right here. It's exactly the same thing. Okay, but this one I'm gonna call R2. Okay, so this here is amino acid 1. This here is amino acid 2. Okay. So if we want to join them together, we're gonna have to use dehydration synthesis, right? It's the same thing that we've been using in all the different macromolecules. So what we're gonna do is we're gonna remove the OH and then we're gonna remove the H. And that's gonna give me H2O, right? That's gonna give me H2O. And then, okay, you don't necessarily have to draw this next part. You can just watch. Okay, once you remove the H and the OH, then you can create a new bond that connects them, right? So this is called a peptide bond. A peptide bond. Right? Now just watch this next part. Suppose I take another one of these things and I put it right here, right? You don't have to draw it. This one, just watch. Right? Let's put it there, right? Then I can remove the OH and the H again, right? And then I'll be able to form another peptide bond, right? Another peptide bond. So that's going to be like another amino acid, right? R3. And then another peptide bond, R4, so on and so forth, right? So you end up having a polypeptide, right? As opposed to a polysaccharide, that would be a sugar. This is a polypeptide, which is a protein. But both polysaccharide and polypeptide are going to be examples of polymers, right? Anybody have any questions? Okay, let's take a go ahead. Yeah.
Sorry, said each protein molecule can have different number and different kinds of amino acids, right?
Exactly. Okay, so some might have 20 amino acids, other might have hundreds of amino acids. Okay, and the kind of amino acid is going to be different as well. Good question. Okay, let's
Quick question. So
sure.
dehydration of amino acids is how peptide bonds are formed?
That's right.
Okay.
But that's how any of the bonds are formed, right? For all the macromolecules, right?
Oh,
like if you want to
okay.
join 2 glucose together, it's also dehydration
Yeah.
synthesis.
Oh, okay. Okay. So, but for proteins, it's called peptide bond. Oh, got it.
Exactly. Yeah, they just have different names, right? But it's a type of covalent bond. They're sharing electrons. Okay. All right. Let's take a 5-minute break. We'll come back at 1:42. Yeah. See you in a bit. So that's the peptide bond, and that's how amino acids are going to be connected together. Now we are gonna talk about the shape of the protein, the structure of the protein, okay? And there are 4 levels of protein structure. All the proteins will have 3 levels for sure, but the 4th level, sometimes it might not be there, depending on the kind of protein that we are dealing with, all right? So primary, secondary, tertiary, as well as quaternary. Okay, so those are going to be the 4 levels of protein structure. Let's start with the first one. Okay, the first one is the primary structure. And I already kind of, give this analogy out earlier, right? The primary structure is the unique amino acid sequence in the polypeptide. Polypeptide is the whole strand, right? But what amino acids are we connecting together? If we read the amino acid from one end to the other, that sequence is called a primary structure. Think of the color beads. Okay, you have 20 different color beads, and you join them together. If you read the color sequence from one side to the other, that's like the primary structure. Okay, that sequence is determined by your DNA. It turns out your DNA contains the instruction to make these proteins. Okay, it will tell your body exactly which amino acids to connect together. So that's the primary structure. Then we move on to the secondary structure. Okay. So here I have what do you call these things? Pipe cleaners, okay? So that's like the primary structure. We have the amino acid all connected together. Now, depending on what amino acids are being connected together, some of them is actually gonna loop together and it forms a little spiral, okay? So these spirals, they are called alpha helix, okay? Alpha helix. And in another part of the protein, they might, instead of forming the helixes, they might form these zigzag like this. Okay, you see the difference? This is zigzag just going up and down, and then over here, that's like spiral, right? Spiral. I don't know if this prop is helping you, okay, but hopefully it helps you visualize what's going on. Okay, so this here, that's the loop. Okay, this here, it just goes up and down zigzag. So the loop is called the alpha helix. The zigzag is called beta theta sheets. Okay. So a protein might have both helix and sheets, or they might only have helix or just the sheets. Okay. So either one by themselves, or it could be a combination of both. It depends on what kind of protein we are talking about. Okay. So these secondary structure is just referring to how many helix you have and how many sheets you have, and they are stabilized, they are held together by hydrogen bonds of the peptide backbone. Okay, what does the peptide backbone mean? Let me go back to our drawing here. Now you don't really have to draw this, okay? But let me just erase this. If I connect a few more things, okay? And you can verify this later when you have the notes, right? R3, and then I do it again. Again, you don't have to copy it, just watch me do it. R4, and then so on and so forth. It just keeps on going and going. You see that the backbone here is not changing. It's the same thing over and over again. It's like, NCC, 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 NCC. That's the backbone. That does not change. What changes along the way are the side chains, right? Okay, so that yellow part there, that the backbone, that is the thing that is going to be stabilizing the secondary structure. Okay, stabilizing the beta sheets as well. Okay. All right. moving on to tertiary structure. So in tertiary structure, oh, sorry, let's backtrack a little bit first, okay? Mad cow disease. Does anybody know what mad cow disease is? How do someone get mad cow disease? Anybody?
Is it a virus infection?
Okay, virus infection. Okay, good try. Anyone else? Have you guys heard of mad cow diseases at all? Anyone? No? Okay. So for those of you who don't know, mad cow disease is something that you can catch by eating contaminated beef. Okay, so as the name implies, it comes from the cows. So the cows can get mad cow disease. And if we eat cow that is infected with mad cow disease, then we can also catch mad cow disease. It is not a virus. It is not a bacteria. It's not a parasite. It's not even alive. It's an infectious protein. Okay. What it does is it will leave holes in the brain and the person will die. So there is this protein called it's called the scrape P protein, or PRPC in this case. Okay, that's the short form. That's the normal version of the protein. And you can see in this diagram that it is going to be a lot of loops, okay, a lot of alpha helix. Okay? And what happens is when the cow has the mad cow disease, the protein is misfolded and then it becomes this form, which has all the sheets, right? Like the alpha helix has become the beta sheets, okay? And this is the scary part, right? When the protein misfold, and if we ingest it and eat it, it doesn't matter if you cook your food, right? Like this is not bacteria, you cannot kill them. Even if you cook the food completely, the protein can still get into your body. So let's say this is a normal protein and this is a bad protein, the protein that has been misfolded, okay? So what happens is the bad one is gonna come and grab onto the good one, and then it's gonna make it bad as well. So now you have two bad ones, and then they will go and find other ones to convert, right? So it's like a bad influence or like a zombie apocalypse. It just keeps on converting all the good proteins into the bad one. And then the bad one will clumped together and then it start to kill off neurons in the brain. And that's why there's going to be holes in the brain and the person dies, okay? So this is related to the secondary structure of the protein, right? Just by switching it from alpha to beta when you are not supposed to, that's going to cause disease in some cases. All right?
Professor, excuse me, it's not any treatment? Someone
Mad cow disease, I mean, like once they found out you have it, then yeah, I mean, they probably can treat some of the symptoms, but then the disease is not gonna go away. Okay, like yeah, it's quite scary.
Okay, thank you.
Professor, sorry for that noisy, but my question is, mad cow disease, what is the cause?
What is the cause of mad cow disease? Yeah,
Yes.
there is some because the folding, right, of the protein is dependent on the amino acid sequence. Okay, so if the sequence changes, then that causes them to misfold, right? Okay, so that's how it happens in the cow. But how people get it is if we ingest contaminated cows.
Okay, thank you. Yeah.
Yeah. All right, so that is that. All right, moving on to tertiary structure. In the tertiary structure, that's the next level, right? So now I'm back to my
John, just
Yeah.
a quick question. Are we
Yeah.
gonna have all that in the tests?
Are you gonna have all that in the test? Yeah, unfortunately, yeah, yes. Everything in the PowerPoint slides and the study guides, they are materials that you need to know for the quiz. Yeah.
Wow.
I know, right? But which is why I'm telling you, it's okay to use your notes. Okay, like memorize as much as you can. Okay, remember as much as you can. But it's okay to have like a help sheet on the test, right? Okay. Don't have your whole binder. You won't have time to flip through everything. Okay. But you know, write down the things that you have trouble remembering, and you know, use it as a quick reference. Don't worry. Okay. Like statistically speaking, people usually do okay in the course. It's only people who don't come to class, or you know, they don't you know, study, then they tend to do poorly. But the questions are very reasonable. Okay. Where was I? Yes. So this is the secondary structure, right, with the helix and the beta pleated sheet. The tertiary structure is the one where it is going to come together. It's going to fold up into like a ball or something. So it's like a 3D structure now. Okay, it's going to fold up into a 3-dimensional structure. And these kind of structures are going to be stabilized by the interaction of the side chains. Okay, so things that are hydrophobic, they might come together. Right? Things that are oppositely charged, they might come together. So it creates this 3-dimensional shape. And finally, some proteins will have an additional layer. That would be called the quaternary structure. Not all the proteins are going to have this. Some of them will. So you will have one polypeptide and then another polypeptide, and they will come together, and together they form a functional protein. On their own, they don't do anything. They have to be together. So which polypeptide you have to bring together for it to work, that's the quaternary structure. Okay, quaternary structure. And these are stabilized by interaction between the polypeptide chain. So this molecule here is something that we will come back to several times in the course. This is called hemoglobin. Does anybody know what hemoglobin does?
Travels oxygen.
Transport oxygen, that's right. So this particular molecule contains 4 polypeptides. So 1, 2, 3, and 4, right? And that would be the quaternary structure. It is very important for proteins to maintain their particular shape. Okay? The shape of the protein is called the conformation. Conformation. Okay. There's a key and log relationship between the protein and its shape. If you change the shape of the protein, you will likely alter its function. Right? Think about like bending the key, it will no longer fit into the lock, right? So if you change the shape of the protein, it will no longer be able to perform its normal function. When you change the shape of the protein, when you break down the protein structure, we say it is denatured. Okay, denaturation. And there are different factors that could denature a protein. Temperature, for example. If you think about cooking an egg, where are the proteins in an egg? Is it in the yolk, or is it in the white? Yeah, in the white, exactly. So the egg white will change from translucent to white. Okay, so that is actually denaturation. The protein has been broken down. It's an irreversible process. You cannot go back, right? Dropping the temperature is also going to denature protein in some cases. Can you think of like a dessert that you can make at home, that you put it in the fridge, and then it goes from liquid to kind of solid? Do you guys know what I'm talking about? Jello, exactly, right? So Jello is protein. I mean, it's a lot of sugar, but there is the protein in it. The gelatin in the Jello causes it to solidify, right? That's basically protein being denatured. Okay, cooking meat as well. Meat has a lot of proteins, right? That is another example of denaturation. Changing the pH can also denatured proteins. That's why if you add vinegar to milk, the milk start to clump together, right? That's pH changing the milk proteins, changing the salt concentration, changing the type of solution. All these things could alter the shape of the protein and therefore cause it to not function. Now, earlier I showed you a picture of hemoglobin, right? Hemoglobin is found in red cells. This is what normal red cell looks like. These are normal red cells. They are circle in shape, and they contain normal hemoglobin. So the hemoglobin protein has 146 amino acids. Okay, so the primary sequence is like valine, histidine, so on and so forth, right? If your sequence is changed at number 6, if it changes from this thing called GLU to another one, and this could happen because of mutations in your DNA, okay? Then the hemoglobin is going to be misfolded, and that will cause your red cells to change shape. You see these red cells? That looks like a moon crescent, right? So these are not healthy looking red cells. We call this sickle cell anemia. Some of you might have heard of this before. People who have sickle cell anemia, they suffer from fatigue, they're tired, shortness of breath, and in some severe form, it's quite painful as well because these red cells get stuck in blood vessels and it cause pain. Okay, so that is an example where if you change the protein structure, then the consequences, it's very severe. Any questions before I move on? Okay. Functions. What do we need proteins for? Proteins are important for forming various structures. They form muscles. You have two main proteins in the muscles. Nails, horns, hair, spider webs, all these things are made of proteins. They also make hormones. We already learned about the sex hormone, but those are lipid hormones, right? A lot of hormones in the body are protein hormones, like insulin. Have you guys heard of insulin? What kind of disease require you to take insulin?
Diabetic.
That's right, diabetes, right? So the type 1 diabetes require insulin shots. Proteins are also energy source, but not the preferred kind. You prefer to burn carbohydrates. If you're out of carbohydrates, then you burn fat and protein as like backup resources. Proteins are also important because they form something called enzymes. I will explain what that is in the next slide. Next slide. But first, I don't know if you guys know this, but there are poachers who hunt down rhino, like rhinoceros, and they cut their horns off, okay, and then they sell it on the black market because some people believe that they either have medicinal value or they it's going to increase one's sex drive. And same thing for the pangolin, right? Some Chinese herbal medicine believe that the scale actually has potent medicinal value for treating things like asthma and stuff like that. So these animals are either endangered or they are on the verge of being extinct because so many people are hunting them down for these purposes. However, based on what I just told you, things like nails and horns, they are just made of regular proteins. In fact, the kind of protein that makes up your nail is the same kind of protein that makes up the horn and the scale. Okay, so there's nothing special about these structures. Okay, like if you believe, you know, the rhino horn is gonna drive up the sex drive, then your fingernails should do the same thing. Okay, but of course it doesn't, right? Okay, so you know, people are trying to educate the public about these kind of things. You know, and once you understand it, then you know maybe maybe we can save some of these animals. Okay, back to enzyme, right? Enzymes is one of the things that can be formed by proteins. I actually told you about enzymes last class. I don't know if you remember, but you don't have to go back. I can go back on your behalf. Just give me a second here. All right, right here. Oh, actually, it doesn't let me do that. I have to physically go back. So last class, I was telling you about the lactose intolerant, right? Yeah, people who are lactose intolerant cannot break down lactose because they are missing the lactase enzymes. Okay, so enzymes are actually these proteins in the body that helps you carry out chemical reactions. And these could be different kind of chemical reactions. They could help break down things, they could help build things, but every reaction that takes place in the body needs to have an enzyme. Okay? The reason for that is because reactions, in order for them to happen, you actually need to supply some energy. And usually that energy is too much. Your body is not going to be able to afford that kind of energy. Okay, so that that energy that needs to be supplied, we call that EA right here. Okay, so EA is the activation energy. All right. So normally it's a lot, but with the help of an enzyme, you lower the activation energy, okay, by lowering the energy required to start the reaction. So think of it as like a scholarship. Instead of paying, you know, $2,000 for the program, with the help of the enzyme, with the help of the scholarship, you can still enter the program, but now you only pay $1,500. Okay, so instead of carrying out the reaction with high energy requirement, the enzyme lowers the energy requirement and you are able to carry it out in the body. Okay, so that's what an enzyme is. Now, to give you an example, here is an enzyme. So an enzyme is a protein and it has what's called an active site. The active site is basically where the ingredients, right, are going to fit in. Okay. Fancy word for ingredient is substrate. The ingredient in a reaction. You know, I know some of you are saying there's a lot of material here, but I want to let you know that, you know, the pre-health program is standardized by the ministry, right? So even if you take pre-health at another college, they will be teaching you the same thing, okay? So I am not the one who's making you do all these things. It's, again, standardized by the ministry. So here we have an enzyme and there's the active site and these are the ingredients. These are the ingredients. So, you know, let's just say this is a glucose. And then let's just say this is a galactose. So they will fit into the active site and then the enzyme is going to do the reaction. They're going to connect them together and then it will split out the product. What kind of product is this? Glucose and galactose. What's the product?
Maltose.
Okay, so there are a couple of answers here, but lactose is the correct one. Okay, you see,
Lactose.
got lactose. Okay, so that's what happens. Okay, that's how you make lactose. Now the opposite is true. If you start with lactose and you want to break it down, let's say you just had some yogurt for lunch, you're gonna break down the lactose. The enzyme will do it for you, and then you will get the product. If you don't have the enzyme to do it, then the lactose will stay undigested, right? And then you won't be able to break it down. That's all. Sources of protein. Right, so meat is an excellent source of protein. For vegetarians or vegans, you can get your protein from beans, soy, nuts. They are all rich in proteins. Okay. We have a few poll questions here, but we will take. You can try them on your own first, and I will take it up on Monday because I want to finish teaching the lesson first, so you can start studying for your quiz. And as part of your study, you can do these questions, and then I will give you the answers on Monday's class. Right? The quiz is not until Wednesday, so we have some time. Okay? So now let's move on to our last part here, which is the nucleic acid. Okay. So this is a picture of your DNA. They are black and white in reality, but we can use special dye to color them, and this would be useful for certain application. But we'll talk about it another time. Chemistry of nucleotides. It's made up of the triplet again, the CH and the O. There is going to be some nitrogen and phosphorus. Okay, so remember, there are 5 key things that makes up 96% of the body, right? It turns out all of those 5 things are going to be in nucleic acid. The monomer, the building block for nucleic acid, is nucleotide. So you probably want to write a summary sheet for yourself, right? Carbohydrate, the building block is, glucose, fructose, galactose. For protein, they are amino acid. And now for nucleic acid, the monomer is nucleotides. Okay, nucleotides. There are 2 main types of nucleic acid. DNA, which is the one that most people have heard of. It stands for deoxyribonucleic acid. RNA, most people probably have not heard of that before the pandemic, unless you study science. But after the pandemic, people know about the RNA vaccine, right? Like the Pfizer one, right? The Moderna one. Those are RNA vaccine. So RNA is another kind of nucleic acid, and you have it in your body. That stands for ribonucleic acid. Okay. Now here is a picture of a nucleotide. And nucleotide has 3 parts to it. There is a phosphate. There is a sugar. And then there is a nitrogenous base. Nitrogenous base. So we see the phosphate here. We see the sugar here. And then we have the nitrogenous base. Now this here on the left side, this is an RNA nucleotide. Whereas on the right side, it is a DNA nucleotide. Now, I'm never going to show you a picture and ask you to identify which one is which, okay? But if you take a look at the sugar here, if you take a look at the sugar here, they look exactly the same, right? The red and the blue. But over here at this particular location, the blue sugar is missing an oxygen. That's why it is called deoxyribose. It's missing an oxygen, right? Okay, so even though they both have a sugar, the one in DNA, it's missing an oxygen, so it's a deoxyribose. Now, when it comes to the nitrogenous base, there are 4 different options. A, G, C, or U for RNA, and then A, G, C, or T for DNA. Okay. Your entire genome, all your DNA is made up of only 4 letters, A, C, G, or T. Okay. In the case of RNA, there's no T. Instead, we have U. So
Excuse me, Professor. What they stand for example, A, G, C, or U?
It's right here, okay? So A stands for adenine, G stands for guanine, so on and so forth, okay? But for now, just, know the letter is fine. Now, some of these nitrogenous base are gonna have 2 rings in their structure. Some of them is gonna have 1 ring. Okay? The ones that have 2 rings, we call them purine. Okay, and that includes A and G. You can remember it like this. Remember the phrase pure as gold. So purines are A and G. Everything that's not purine, they are pyrimidine. Pyrimidine. Okay, so purine has 2 rings. Pyrimidine has 1 ring. Okay. The DNA structure is a double helix. It's a double helix, meaning they are going to basically have 2 strands. Think of a ladder. Okay, think of a ladder like this. And then you twist the ladder up. That's what a DNA structure is. The side of the ladder is what it is made up of the sugar and the phosphate. Remember the nucleotide, right? It has the sugar, the phosphate, and the base. Okay, so just the sugar and the phosphate, they make up the sides of the ladder. The rugs, like these ones that you climb on, that is made up of the nitrogenous base, the base. Okay, so you can see the A's and the T and the G and the C, right? So from this picture, you might be able to see that G and C always goes together, and A and T always goes together as well, right? So that's called a base pairing rule. If you know one side is A, the other side has to be T. If one of them is G, the other one has to be C. Right? We are talking about DNA right now, so there is no U, there's no uracil. So this property where you can match them up, that's called complementary. Okay, these base pairing rule, that's called complementary. Now, if you look at this picture, you can see one strand is upside down compared to the other, right? So that's another property. Don't worry too much about it right now. We're gonna learn this again in lecture 6, but this is called anti-parallel. It just means they are parallel to each other, but they run in opposite direction. Think of the highway. You have a northbound 404 and a southbound 404. The 2 roads are parallel, but the traffics are going in opposite direction. That's what anti-parallel means. Okay, I know it's almost time, but I just have 3 more slides I want to go through with you. If you need to go, that's okay. You can watch the recordings, but if you could say, that's fine as well. So let's practice this together. Find the base on the other strand. If one side of the DNA is A, G, G, C, so on and so forth, we can actually predict the base on the other side using the base pairing rule. Okay, so what does A always pair with?
T.
That's right. It says right here, right? A is always going to go with T. So T. G is always going to go with
C.
C. Right? So C again, and C goes with G. And using the base pairing rule, we are able to complete the other side. So that's one strand of the DNA, and then that's the other strand. They are complementary. If you know one side, you can predict the other side. Okay. Now remember, I was telling you, purine are A and G, right? Pure as gold. Purines are A and G. These are two rings. Pyrimidine, that's the T and the C. Those are the ones that have, if you look back, one ring. So look, purines always pair with the pyrimidine. Look, A and T, right, are together. A has two rings. Thymine has one ring. C and G. G has two rings. C has one ring. Okay. So if you check it across the whole strand, between the two sides of the ladder, it is always three rings across. And that's important for stability. Think about it. You want a ladder that's uniform in width, right? You don't want a ladder that's like bulging out for some parts and then, really narrow for another part. Like this is not stable. Right? That's why it's important for the purine to bind with the pyrimidine. It's always the same distance across. Two more slides.
Excuse me, Professor, about the purine, always
Pyrimidine, yeah.
A and G is purine.
That's right. Only A and G are purine. That's right.
Why A and G, Professor? Because A,
Because
sometimes
A, because,
they are the same line,
no,
sometimes they are opposite.
okay. They are called purine because they have two rings. That's why.
Because
Okay.
of the rings.
That's because of the rings. Yeah.
Okay. Okay, thank you.
Yeah. Suppose a cell has 40% adenine, and we have to find the percent for the rest of them. Okay. So 40% A. What percent of T do you expect? If you have 40% A, what percent of T do you expect?
40%.
40%. That's right, 40%. It's the same, right? Because whenever you have an A, you're going to have a T as well, right? Now you add these together, that gives you 80%, right? Total is 100, right? So 100 minus 80, we still have 20% of stuff. What stuff are we talking about? I already account for all the A and T. So what is the rest of this stuff?
C and G, 10%.
Okay, C and G, that's right. Okay, and of course C and G are the same. So if you divide by 2, you would have 10%. Exactly. Okay. If you didn't understand that, that's okay. Watch the recording. And then, we can talk about this again next class. Last slide. The other kind of nucleic acid is the RNA. They are different from DNA because they are single-stranded. It's not two strands, it's just one strand. And the kind of sugar they have, it's the one that has the oxygen. Okay, we call that the ribose sugar. And instead of thymine, they have something called uracil. Okay, they are pretty much the same thing, but it's just like a replacement. Okay. 3 types of RNA, they are listed here. We will learn much, much, much more about these things later on in the course. I just want to put it here to show you that there are different kinds of RNA. Okay, So I know there's a lot of information. I thank you for your patience and attention. There are all these practice questions that you can try. I will take them up next Monday. If you have any question or if you need extra help before the quiz, please email me. We can, I can help you through the email, or we can set up a time to meet virtually. Otherwise, have a good rest of your week, and I will see you on Monday again. Thank you.
Thank you, Professor. Bye.
You,
Bye-bye.
Professor.
Yes.